So I'd like to welcome everybody today. We have the privilege of hosting Professor Patrick Wong, who is the Stanley Ho Chair in Cognitive Neuroscience at the Chinese University of Hong Kong. Prior to joining CUHK 11 years ago, he was tenured at Northwestern, where he started his faculty career. He is a cognitive neuroscientist, linguist, and a speech-language pathologist. And he's gotten quite a lot of recognition, both from the media and in the form of grants. Today, he's going to speak to us about Predict-to-Prescribe approach to enhancing children's language development. And let's welcome him. Thank you all for joining us.
Thank you for the introduction. So I'm going to talk about language development in children. So one thing that we know about language development is that it's actually quite variable across individuals, across learners. And so here I have three different examples of the learning trajectories of three groups of children from three different studies. Each line on each graph represents a child's development. And as you can see across tense marking, vocabulary development, and whatnot, you can see quite a bit of variability. So some children may learn to be like this, but there are children who are more like this. So there's quite a bit of variability. Now, from decades of research into development, we know that development can be quite stable. So we call it developmental stability, and language is not alone. And so this is what I mean by developmental stability of language. So let me give you an example of 5 children using this graph here. So child A, B, C, D, and E. So pretend that this is their ranking when they're at age 2. So A is worse than B, B is worse than C, E is the best scoring child on this graph. Now, when we say that they're stable, we mean that at age 7, for example, when they're older, everyone improves. However, the ranking stays pretty much the same. So at the group level, what we know is that children who tends to be low to begin with, they continue to rank low. Children who rank high to begin with, they continue to rank high at the group level. And so we have a lot of data showing us that. So language, importantly, language for the purpose of our talk here. Across populations, we see that. So children who are hard of hearing or children who are born preterm, they show developmental stability. And interestingly, how parents communicate with their children is also stable. So parents who talk a lot to their children continue to talk a lot to their children, okay? This is actual data, a relatively recent study by Bornstein and his group. So they looked at the Avon longitudinal data from the UK, from over almost 1,000 children. So 6-month communication can predict 1 year, can predict 2 years, and can predict all the way to year 15. So if you know something about the child's language and communication at age 6 months, you can predict the child's language development at age 15. Now, this is group level results. This is not to say that if I pick a particular child, if I say Johnny, measure Johnny's 6-month communication ability, I can like say that, oh, Johnny is going to get an A on the Hong Kong SAE, DSE. Okay, so we're not talking about that level of precision. It's not individual level prediction. This is group level prediction. But I will talk about individual level prediction throughout this talk. Okay, now I study language, and so language impairment is something that many parents are interested in and concerned with. And so we know that language impairment is linked to a whole host of future development, including behavioral adjustment and potentially future mental health, academic achievement, a lot of parents are concerned with. When you look at the DSM-5, we know that the majority of neurodevelopmental conditions are associated with some form of language and communication problems, including autism. So that's why a lot of parents care. And what we're interested in, so throughout this talk, I'll come back to this point, is that we're interested in making prediction. So we're interested in making prediction based on data that we collect early from the child and use that data to predict developmental trajectory. Okay, and we're particularly interested in collecting data in infancy and toddlerhood to make prediction about preschool years and beyond. Why do we like to predict? Now, going back to the concept of developmental stability that I just mentioned. So if development is stable, it means that data that we collect early can tell us something about the future, right? So if we can forecast early, then we can find ways to intervene as early as possible to take advantage of that window of neuroplasticity. So, we have economists telling us that, from their analysis and simulations, Heckman, for example, telling us that the return on investment is higher if you try to intervene early. If you wait till school years, you have to put a lot more resources into getting the same amount of outcome. And then from neuroplasticity data, we know also that if we try to change the brain early on, it's easier than try to change the brain later on. And therefore, if we can predict early, we can then intervene. Okay, so this is the overview of my talk. So first, I'll show you some data from a community sample of infants and toddlers. We measure what we call the neuroencoding of speech, and then we use that as a marker for future language development. And then I'll talk about using neuroencoding of speech to make prediction about not autism diagnosis, but early intervention, how much the child respond to early intervention, okay? And then if I have time, I'll talk about some MRI data that we have. We use MRI before surgery to predict post-cochlear implant surgery outcome, language outcome of children at the individual child level. And then if I have more time, I'll talk about our latest study using anatomical MRI to forecast language outcome in preterm infants. So about 10% of children are born preterm. And so if you look at their developmental trajectory, we see that there are a lot of kind of hidden problems that we don't detect until the child is 5 or 6. And so therefore, making prediction early is very important from the intervention standpoint. So the title of my talk is Predict-to-Prescribe approach. So let me tell you what we mean by that. So we want to first predict, and then we use prediction results to inform about prescription of intervention. So what to predict? So I mentioned a little earlier. So we want to predict early using neuro and non-neuro data to construct predictive models to forecast developmental outcome, using data from as early in life as possible, so infancy. In fact, now we're doing a project looking at neonates, okay, collecting EEG data from neonates. Another way that we want to predict, another measure that we want to predict is not only developmental outcome, but also how well the child respond to intervention options. So there could be different ways of intervening, and we want to use data to inform which ones of the options of intervention we would like to prescribe. So prediction here has two meanings. One is to predict developmental trajectory, another one is to predict response to outcome. And then based on our predictive models, we want to prescribe early intervention. So if the child has an elevated likelihood of language and cognitive difficulties, then we want to prescribe early intervention as soon as possible. We don't want to wait for a formal diagnosis. Okay, we want to act right away, and then we can talk about what those intervention options might be later on in my talk. And we want to be able to prescribe the option of intervention that would most likely result in the most optimal outcome for that child. Not for another child, but for this particular child. So individual child level prediction is what we're trying to aim at. Okay, so let's talk about a sample of community-based infants and toddlers that we've been studying. So I'll be talking about neuroencoding of speech. And so I want to give you a little bit of background of what I mean by that. So it's going to be neuroencoding of speech. So I want to talk a little bit about speech perception. Now, when we think about hearing and speech perception, very often we think about the ear, right? Because the ear is the organ for perceiving incoming information. Now, a lot of times people don't think about is that the auditory signal actually eventually arrives at the brain. Right. So the brain is actually involved in the hearing process as well. More specifically, I mean the central nervous system. So including the cortex and also brainstem nuclei. And neurophysiologists would tell us that when we go up this auditory pathway from the ear to the auditory cortex, we can see neurons that are sensitive to increasing complexity of the auditory signal. So for example, if you look at neurons in the periphery, you can see neurons that are sensitive to like high pitch or low pitch. When you go to like the inferior colliculus in the rostral brainstem, you can see neurons that are sensitive to frequency modulation. And then when you move up even higher to the brain, to the cortex, you can see neurons that are sensitive to speech and music. Okay, so we have this kind of hierarchy of complexity, functional complexity, as well as acoustic complexity. Now, when we think about speech, very often we think about the cortex, the auditory cortex here. But what we know is that the auditory pathway actually passes through many different nuclei that are subcortical. And subcortical nuclei actually receive connections from bottom up and top down as well. So it's actually a very complex kind of picture. And a structure that we are very interested in my lab is the inferior colliculus, for reasons that I'll explain to you in a bit. Now, this is the area of the brain that we can collect neuroencoding signal. We can look at phase locking to the frequencies of the incoming speech. And so that's why this is an area that we pay attention a lot. So in my lab, we use this paradigm a lot. This is called neuroencoding paradigm. Basically, what we do is that we present a speech signal to the listener like this using EEG. And then our active electrodes here is collecting how the neurons phase lock or respond to the incoming speech signal. So you can see that this wave here, the second wave here, is the wave that represents what the neurons, how the neurons actually respond. Okay, so you can see that the brain wave is actually very closely resembling the sound wave that we present to the subject. Okay, so that's why we call it encoding. It's encoding for this adult subject very faithfully to the incoming speech signal that we presented to her. Okay, so I'm going to play you two examples. So one low pitch, one high pitch. So this low pitch sound, you can see the waveform. This is the waveform coming out from this person's inferior colliculus. And this is high pitch. You can see the resemblance. So, but let me play you the signal that we played to her. And then I'm gonna play you this signal, which is what we collected from this subject's auditory system. High pitch. Okay, can you hear the resemblance in pitch? Okay, so this is a good audience because not every audience gets it. Yeah, but so thank you. So let me do it again. Okay, you can actually hear what the subject heard. Okay, so this is neuroencoding. And so in a lot of our studies, we're interested in this neuroencoding signal. Now, this is basically an upgraded version of newborn hearing screening. So many of you may know, for those of you who have children who were born in Hong Kong, they all, if they were born since a few years ago, they all went through this newborn hearing screening using what they called a click ABR. So basically, you present a click sound to the child to see if the inferior colliculus responds. Okay. Now, our test is an upgrade of that because we use a speech signal and plus all the AI and all that analysis techniques that we use. Okay. So, and I'll come back to this because our test is actually being commercialized. So the trade name is Precision Listening. Okay. And I'll disclose my financial interests in a bit towards the end. Okay, so we have some examples here of infants and adults processing the rising tone, encoding the rising tone. So the black line is the pitch contour of the pitch of the speech sound that we present to the subject. So this is a group of infants and this is a group of adults. You can see that the adult, on average, this is average across a group of adults. You can see that the signals look very well. Infants is actually not bad, but not as good as adults because their nervous system, central nervous system is developing. Now, this technique that we use is very important from the clinical standpoint because the signal is actually interpretable at the individual subject level. So even though we do group average, I can pull out the spectrogram of or the waveform of an individual infant and interpret that signal. And it's very replicable within individual as well and across environments. Okay, so that's why this is being used for clinical purposes, and then our upgraded version is based on that. And so that's why for the purpose of individual level prediction, this is something that we need. So many of you may be familiar with other EEG paradigms like the MNN. With MNN, for example, you cannot just pull out the MNN from the individual subject and make interpretation. Okay, so but this technique is interpretable at the single subject level. Now, some infants are better than others. So these are two examples of two individual 4-month-old infants. Okay, we presented the same signal to these two babies. This one, the nervous system is noisy. Okay, you can see that it's kind of fuzzy, but this one is encoding to the pitch of the stimulus that we presented to that. Okay, so this is a lot clearer. And so we've been conducting longitudinal studies. We follow maybe 600 or 700 children by now. So we follow them for up to 5 years. And now we know their results, their developmental results, right? And so for these two children, this particular child, their language measure is at 10 percentile, and this one is at the 99th percentile. Okay, of course, I'm pulling just two subjects. We have a group of subjects already, and I'll be showing you some of the results. Okay, so what we've been doing is we've been collecting data like this, and then we're doing longitudinal studies, follow a group of children through kindergarten, and some are in primary schools already. Based on the data that we collected, we use machine learning models to construct predictive models to predict their future language development and language and cognitive development in their kindergarten years. Okay, I'll show you data in just a minute, but that's the concept of it. And so now when we encounter a new child, we can use the same EEG paradigm. We put the EEG data into our algorithm, and then we make predictions about the child's future language development. These are some of the neuroencoding features that we measure, because sometimes the audience asks. So it includes what we call measures related to myelination, like speed, and then measures related to dendritic processes. Those are measures that are related to like the connections of the neurons. So there are different signal processing that we have to do. Now, this is some results from our first study. So again, we took these neural measures, and then we also have non-neural measures. So what are non-neural measures? Like birth weight, gestational age. So we know that preterm babies, their language development is not as good as full-term babies as a group. We also know that babies who are born really not very heavy, okay, like light babies, their development is not as good. Okay, so we can use those clinical measures to make predictions about their future language development, right? Now, so let's compare these two types of data, neuro data and non-neuro clinical data, okay? So if we use clinical data, like birth weight and gestational age, to predict future language abilities, okay? Using a binary classification, 16 percentile is the cutoff. 16 percentile is one standard deviation below the mean. Using a binary classification machine learning models, if we use pediatric factors like birth weight and gestational age, our AUC, which is a combination of sensitivity and specificity, is about 60-some to 70%. It's significant, okay? But 70% is not very useful clinically. Now, once we add the EEG data, add the neural measures, our AUC is at low 90s. Okay, this is when we start to be clinically useful. Okay, now this is our first study from 118 babies. Now we have over 500 babies. I'm going to skip that. So, again, that's our first study. Now we have more babies, and so we have more data to see if our model is still accurate. So this is data from 300 babies from 3 weeks old to 24 months old at the age of acquiring the EEG data. We follow them for a while. So our language outcome here is the CDI, the MacArthur CDI is a survey that parents fill out about, like we ask parents whether or not the child knows these words. The child knows these words, okay? Whether or not the child can comprehend, whether or not the child can produce those words. So that's basically what the CDI is. It's been used extensively in research for decades, validated across different languages, across different clinical populations, okay? And so with 300 babies, our AUC is still at 92%. Sensitivity is 83%, which is comparable to a lot of different clinical measures. For example, mammography for breast cancer is 80-some%. Okay, now this is CDI. Now, when I talk to pediatricians, they were like, well, CDI is not a real clinical measure. CDI is just a survey. Even though you keep explaining to them that, it's been validated on the various different measures, they still don't believe it. And so then what we did is to collect more data. And then we use Bailey as the outcome measure. Okay, so the Bailey is an instrument that's a comprehensive instrument that you directly assess the child, okay? We measure the child's language and cognitive. We're using cognitive performance. We use EEG again to make predictions across these two domains. Our AUC is still pretty good. Okay, it's 87% and 83%. So language is better than cognition, but it's still pretty good. It's in clinically useful realm. Now, clinicians and pediatricians also ask these questions. They ask, like, why do we need EEG? We can just use pediatric data like gestational age and birth weight to make prediction, right? Yeah. And I told you, yes, but it's just not very good. I mean, it's significant statistically, but it's not very good. And then they also ask, well, then maybe use some other instruments. For example, the ASQ. For those of you who do developmental research, you're familiar with that. The ASQ Ages and Stages questionnaire is a questionnaire that parents fill out, use extensively across different populations. It's basically a checklist that the parents like check, or 1, 2, 3, there are different categories, and then you add up the scores, and then you tell the parents that, oh, your child is on track developmentally. Okay, so you pay $10 US, and then you can access the scale. It's very cheap, and so pediatricians will ask, why not just use that scale to make predictions about future language development? So we did. Okay, so ASQ is actually not very useful. You can see the distribution here. It's like 55% AUC. Pediatric factor, I told you already, always about 70-some%. EEG alone is 93% per our model here. So there's something about the brain. The brain is, I explained to parents, the brain is the learning organ. And therefore, when we measure the brain directly, we get more accurate data because that's like the more direct means of looking at how well your child is going to learn. Okay, now this a pitfall of machine learning. Like I'm sure like all these AI stuff now, like everyone is using AI. Now, a lot of times there's the problem of overfitting. So the models look really good when you applied it onto your own data. Like if you use the data that you collect to build a model and then use the same data test set to test your model, your what we call internal model, you may be overfitting. So what we needed to do is to use a brand new set of data that's not involved in training to test our original model. Okay, so we use random forest here, use a brand new set of data to test our original model. It still works. Okay, sensitivity 90%. Okay, so AUC, both AUC and sensitivity is at 90%. Okay, so this is very encouraging. Now, the internal model with like now a lot more children is 97% AUC. The external model is not, the external data validation is not as good because it's external. Okay, but it's still within a really good range for clinical usability. Okay, now, because we're collecting these data very early on in life, infancy, then people may ask, like, why not just look at genetics? Like, so are you saying that this is genetic? And if it's genetic, then why can't we just like do a little swab of the child, and then like just look at the genetic data and then try to use prediction? It's a lot faster and cheaper. You don't have to go through, put the electrodes on and buy the expensive EEG machine and whatnot, right? Okay, so we do conduct genetic experiments in my lab. I'll show you two studies. This one we published 4 years ago now. We looked at a genetic variant called ASPM. In particular, this is the position that we looked at. I can explain to you why we looked at this area because of previous research suggesting that this is linked to Chinese lexical tone perception. So we looked at this variant to see if it's linked to Chinese lexical tone. In fact, it is. So we achieved statistical significance, okay, based on a sample of 426 subjects that we collected, this particular genetic variant, right? Now, but the problem is this. We're only explaining 1.4% of the variance in the data. It's statistically significant, but it's very tiny. Without a large sample size, is not going to show up. So from the clinical individual child prediction standpoint, it's basically useless. Okay, we publish these results. That's why I can criticize myself. Okay, so these are results that we publish. And, we got into Science Advances because we found the association, not because it's useful. Okay. Now, we also did another study in which we looked at all the genes that are genetic variants that have been previously reported to be related to language phenotypes. Like FOXP2, for example, catnap. Those genes have been associated one way or another with some form of language phenotype. Like non-word repetition, comprehension, and whatnot. So we collected all of those and put them into the same platform for analysis. And our phenotype here is, so these are all Chinese university, mostly Chinese university students. So we have their DSE English score. So their DSE is the public exam for those, I'm sure you know what that is. So we know their second language English score, right? So we were trying to see if we can, we can use the genetic information that we collect and make prediction about their DSE English score, right? So lo and behold, when you have a sample of 400 subjects, these are the genetic variants that show significance. Okay, where's my cursor? Can you see? Well, okay. These are the genetic variants that show significance associations. From the same subjects, we also collected information about their music background and their family socioeconomic status. These are the variants explained, okay? So you can see that these are the significant results. So these genetic variants collectively only explain no more than 2% of the variance. Okay. Now, the interesting thing I'm just going to say interesting thing is this. If you look at music alone explains 2.6% of the variance, already more than the genes, all the genes combined. This sad thing is this. Look at family socioeconomic status. It explains 5% of the variance. It explains the most variance is how much money your parents have. Okay, in terms of how well you do on your DSE. Okay, so again, 421 subjects, genetics, there's significant genetic association. It's just that it's not very useful. Okay, from the practical standpoint, it's not useful. From the theoretical standpoint, it's interesting to see that these genes, actually, we replicated a lot of the results that have been published, especially from samples from English learning, first language speakers. Okay, so my point being here is that genetic is useful. Genetic is part of it, of course, but we cannot rely on genetic information to help us make any kind of decision as when it comes to language. So I'm not going to play you the video, but if you're interested, you can go you can look at that. This is the video is to show you how the test is conducted. So we have this happy baby. They're not always happy, but in this model that they happen to be very happy when we conduct the test. So it's a short EEG test that we administer, and then based on the results, we put it into our machine learning model, and then we make predictions. And so this is patented technology that we have. We won the gold medal from the International Exhibition of Inventions in Geneva about a year ago, thankfully. Okay, so if you're interested, you can go to this QR code and then look at how it's done. It's being commercialized in Hong Kong. Okay, so this is all kind of clinical. So let's come back to some science for a bit. Now, with the data that we collect, we can actually use it to test a number of different hypotheses about language development in the brain. So one hypothesis that we're interested in is Pat Kuhl's native language neural commitment hypothesis. So this is a hypothesis related to how the brain supports language development. So according to Kuhl's hypothesis, the way that language develops is based on how much the brain devotes resources or commit neural resources to processing the child's native speech sounds. If early on the child understands that the brain's neurons should be only pay attention to the speech of the native language, according to Pat Kuhl, that child's language development will be better. Okay, so that's why it's called the native language neural commitment hypothesis. It's early commitment from the child's brain to the child's native language. In other words, from the clinical standpoint, according to Pat Kuhl, if you present the baby with a native speech sound, that sound should predict non-native speech perception better in terms of future language development of the child. Okay, and they have some data from a while ago. So this is a 7.5. In this study, they looked at 7.5-month-old infants in an English learning environment. They use a cortical paradigm called MNN. For those of you who know what that is, it's just a brain paradigm. They presented English sounds to these babies, a bunch of English English speech sounds, and then some Mandarin speech sounds. Mandarin is non-native, English is native. Okay, so at the group level, you can see that the child is actually very sensitive to the native sound. That's why you have a huge gap here. The child were not that sensitive to the non-native speech sound because it's not in the child's environment, right? And so then the next question for them is whether or not these results will predict the child's language development, or whether or not these will predict future language development in the negative direction. So if you're devoting your brain to listen to something that's non-native to you, that should predict poorer language development, okay? Because you're devoting resources to the wrong things. You should be devoting resources to this, neural resources to this, not this, right? And in fact, they have data suggesting that. So children who were better native native perceivers, their language development in this is vocabulary is better. Okay, if they pay attention to the native sound, their language development is not as good. Okay, so now the important thing for us is this. MNN is a cortical response. What we're measuring for the most part is a subcortical response, although we also have some cortical response in it, but for the most part, it's subcortical response. So the question for us is whether or not at the subcortical level, this neural commitment hypothesis is still true. Okay? Yeah, right. Okay, so in our neural encoding speech paradigm, we included 3 speech categories. Tone 2, which is the same for both Cantonese and Putonghua. Tone 4, which is only Cantonese. So this is a Cantonese low falling tone. It does not occur in Putonghua. And then we included the tone 3, the Putonghua dipping tone 3, which does not occur in Cantonese. So in other words, we included both native and non-native speech categories in our paradigm. All of our infants, so here I show you data from 386 subjects, they were all Cantonese learning. In other words, tone 2 and tone 4 will be native to them. Tone 3 would be non-native to them. And so what we're able to do is to construct predictive model to see if the native speech categories predicted language development better than the non-native Putonghua tone 3. Does that make sense? Okay, now these are the prediction models, predictive results. The 3 categories were on top of each other. We did not find evidence that native is better than non-native. This is telling us that probably at the subcortical level, the nervous system doesn't really care what is encoding. What we need to measure is purely the brain's response, like a really raw kind of response to the incoming speech signal. Perhaps the job of the cortex is to help differentiate whether or not this is a native speech category or non-native speech category. Subcortically, we only need to measure how well those neurons respond to speech, regardless of what kind of speech the child is listening. Okay, so I think it's helping us to rethink how the brain processes native and non-native speech and what that might mean to language development. And in fact, we collected some longitudinal data, both cross-sectional and longitudinal data. We looked at development from 23 to 730 days of development. You can see these developmental trajectories. Across different neural measures that we collected, it's pretty linear across both native and non-native speech sounds. There's no break in that in the development. Okay, it's not like when it's non-native, it stopped growing, and then it's only growing when it's native speech categories. Okay. Okay. Now, let me switch gear. So the point here for the first set of study is that we're able to predict. Okay. And we can use the same set of data to test different hypotheses, okay, about how the brain is responsible for language development. So let me now switch gear to talk about autism for a minute, autism in Chinese children. We can use the same paradigm, neuroencoding of speech paradigm, to test children with autism and typically developing children. And we've published these results on school-age children. We can differentiate children who have autism from children who are typically developing using the same techniques. These are this paper here is for school-age children. We also have data on preschool learners. I'm waiting for more subjects. We have 70-some subjects in this poster that we presented. I'm waiting for more data before we publish. Hopefully getting over 100 to get into a better place. So with preschoolers, we can still differentiate using the same technique. Now, what we're interested in this next study that I'm presenting to you is not just about categorizing children into diagnostic categories, but whether or not we can use these neuroencoding results to inform treatment, intervention. Okay, so let's talk about intervention. So how do we intervene when the child is very young? When we're talking about infants and toddlers, I'm not talking about school-age children because our populations now is mostly infants and toddlers. How do we intervene? Now, we know that there's no drug treatment, there's no magic pill, and behavioral intervention is the only realistic option for parents who choose to get intervention for their children. You know, some parents, they just think that it's fine and there's nothing that they need to do. And we respect that. Now, what we know from the literature, from the best evidence, the best way to intervene is through social communication with the parents. So basically, we teach the parents how to communicate with their children so that the parents can implement treatment at home. Why do we do that? Well, social communication and language is not a 1 hour per week thing, right? So, because the child is constantly developing language, the child needs to be getting a lot of the language input. If the child only comes to the clinic 1 hour a week, that's not enough dose. Parents and caregivers are the people who are able to provide that dose to those children. Okay, and therefore, the best evidence that we have about treatment for social communication for infants and toddlers is to teach parents, is to coach parents how to do treatment at home. And we have a ton of data to support that. So this is a meta-analysis conducted by my colleague, Megan Roberts at Northwestern. This was published in JAMA Pediatrics, a meta-analysis of thousands of children. And what she found is that for expressive and receptive language, the effect size can be close to 1 if you use this kind of treatment paradigm. Okay. Receptive language is especially noteworthy because if you look at the data from 4 years of age and on, a lot of data is suggesting that receptive language treatment actually doesn't work very well. And therefore, like catching the children early in the infancy and toddlerhood is very important because it can result in really good treatment effects. So what we did, I'm going to skip this. What we're doing now, this is an ongoing RCT, is to take the best features of parent training and implement it in the local context. So we develop local materials, play routines, and then we're testing whether or not we can prescribe this kind of training online and in a group setting, whether or not it will still work. Okay, we're comparing it one-on-one. And so we're conducting 2 RCTs, one in Hong Kong and one in mainland. We finished enrolling already. Now we're just doing follow-up. So a therapist, when we first proposed to do this, we were going to do it in person. And because of COVID, we were forced to do everything online. And now we're finding out that actually it works to do it online as well. Okay. And so one therapist would treat up to 8 families at a time online. And we are finding moderate effect sizes on our primary outcome measures so far. Okay. Now, but this is what I want to tell you. Even though at the group level, when you look at the RCT results, at the group level, it works, right? We know that it works. Locally, it also works. But we still see quite a bit of variability in treatment response. Okay, so let's look at here. So each line here, I hope you're used to looking at these graphs by now. Each line represents a child's trajectory, a child's, in this case, receptive communication. So this is before treatment. This is after treatment for each child. Okay. So we have, I don't know how many, I have 50 some children here in this preliminary analysis. You can see that they're all over the place. There's quite a bit of variability in response to intervention. Okay. So this is a child that we want, right? Parents want, is not so good at the beginning, after treatment, huge improvement. Nice positive slope is what parents would want to see. Like this is another child, this is another child that you want, right? Now, some children are the black lines. These are the children who did not improve, they regress, in fact. So treatment didn't really work on these children. Okay, some are pretty flat, like this is not good. Okay, so we like these, right? So if you are parents, what you would like to know before treatment, before intervention, whether or not my child will be more like this child or this child, right? If my child is going to be like this child, I'd rather not go through intervention, or I'll do something else. Not go through intervention, but do some of the intervention. Perhaps this is the child that requires really frequent speech therapy visits. Like maybe they need the therapist to do the intervention and do it all the time. So that will be very expensive. But that's what the child needs, perhaps. But this child, really parent intervention, like just do it online and whatnot would work. Okay. But we want to know this before treatment, before intervention, not after, right? Okay, so how do we do this? So we've constructed different predictive models to predict response to intervention. Okay, whether or not the child would improve or not improve, a binary classification. These are AUC results, again, a combination of sensitivity and specificity. Permutation is the random model, therefore it's at 50%. We can use pediatric factors, gestational age, birth weight. Those are the pediatric factors that we've been talking about. If we use those factors, I don't know if you can see the number, AUC is 60. We use also, we build models using what we call psychological factors. So those will be the developmental measures that we measure from the child directly, like their ADOS score, like indication of their autism severity, for example. We can use that to make prediction. AUC 65%. Now, when we use our EEG data, AUC is 81. These are preliminary results, okay? Just preliminary results, okay? And then we can combine like pediatric factor with psychological factors, PD versus neuro, PD and psych and neuro. When you combine everything, you know, it's no better than neuro alone. So the brain is already a summary measure, basically. You don't need other measures. The brain tells you all that you need to know. Because, for example, if you are preterm, is reflected in your brain already. So you don't need additional information about whether or not the child is preterm. Okay, at least that's our hypothesis. Okay, and so far it seems like using EEG can also help us predict whether or not the child will respond well to the type of intervention that we prescribe to the child. Okay, so we can use neuroencoding to make forecast about future development, and also make forecasts about response to intervention for children with autism. I have 5 more minutes, 10 more minutes, something like that. Okay. So quickly, I want to tell you some. So EEG has, you know, we use EEG a lot because EEG is clinically more useful than MRI. You know, MRI is more costly. And then if we don't want to sedate the child, to scan the child is, you know, there are a lot of issues surrounding it. However, I want to tell you that we are also doing a lot of MRI experiments in children. So this is a study that I've been doing for more than a decade now. This is to look at children with cochlear implants. Okay, again, these curves, these learning curves. So let's look at children. So for those of you who may not know, for children who are born hearing impaired, if they are very severe and cannot benefit from having a hearing aid, the best treatment option now is to provide them with a cochlear implant. But it's a surgical procedures, and it's, you know, it's a really a big deal, and some parents just don't want their child to go through that, or because the child is deaf themselves, they don't see the need of giving their child a cochlear implant. Okay, but here I'm talking about children whose parents make the choice of implanting their children. Okay, which is the vast majority of deaf children. So by now, I hope you're used to looking at these graphs. Each line represents the developmental trajectory of a child. This is a group of children who have cochlear implant. The yellow lines, the gray lines are typically their normal hearing peers. Okay, and so you can see that as a group, children who have normal hearing, their language development is better than children with cochlear implant. Even after the cochlear implant, they're better than their deaf peers, but children with cochlear implant is still not as good because the cochlear implant device is not perfect. What we're born with here is really the best hearing device. Okay, so if you replace it with an artificial device, it's still not as good, but it's still better than having nothing. Okay, if the goal is to develop spoken language. Okay, now when you look at a graph like this, this is from Naparko, I can't remember, published in Lancet, I think. So for me, for my own research interests, it's not only the average that I'm interested in. I'm interested in individual differences. And so you can see that this this fan-like structure, meaning there's a lot of variability in how well children with cochlear implants develop language. A child like this, after the cochlear implant, the child's language is not very good, spoken language, but a child like this develops very well, overlapping, even more, very similar to normal hearing children. If you look at normal hearing children, that distribution is a lot tighter. Okay, there's not as much variability. So in our research, we're interested in predicting this variability. Okay, this fan-like structure. Whether or not before surgery, we can predict whether or not a child will end up being like a child like this or being a child like this. And if we know that the child is going to be like this, we're not denying treatment. What we're going to do is to plan more extensive therapy post-surgery, or even before surgery. We might already be teaching parents how to conduct treatment at home to stimulate their children. So this is our paradigm. We would collect MRI data before surgery. So these MRI data we already collect as part of the clinical assessment. So these are free scans, if you will. In fact, we asked the radiologist to modify the scanning paradigm a little bit so that we can use the data for research. And then we follow the children up after surgery at various intervals, 6 to 12 months. They will come back to the clinic, and then we'll conduct different assessment. And then, so what we do is we use these scans to construct predictive models of their post-surgical outcome. Okay. I'm not going to go through this in detail, except to say that we've been conducting these predictive models based on MRI scans. If you compare hearing children with hearing impaired children before surgery, after cochlear implants, they cannot get an MRI. So this is all before surgery. You already see differences. So if you see colors, that means they are related to hearing impairment. So the deaf children differ from the hearing children in these brain networks. Okay. So of course, a lot in the auditory cortex, but also attentional areas, cognitive areas as well. So with these data, we can start thinking about developing hypotheses about what kind of brain network may be associated with maybe more predictive of future language development. For example, we can think about whether or not areas that are affected by hearing impairment, these color areas, are more predictive of future language development, or actually areas that are outside that are more common between hearing and hearing impaired children that are more predictive. Okay, so there are different hypotheses, and they relate to, thinking about compensatory mechanisms and hearing impairment, and how that might, and what auditory, the lack of auditory input may do to the brain, to change the brain. So I'm not going to go through that in details. I'm going to tell you about the results. So random is in gray, which is about 50%. So we constructed different predictive models using different brain network. So network that are affected by hearing impairment, network that are unaffected by hearing impairment to make predictions. And what we're finding is that brain areas that are unaffected by hearing impairment seems to be the most predictive about future language outcome. In other words, it seems to us that areas that are outside of the auditory areas, typically, children who are more likely to use compensatory mechanisms would be the children who achieve language outcome better. So not try to rely on the hearing areas of the brain, but rely on the cognitive areas of the brain to bypass their hearing difficulties. Okay, so and importantly, we also build models using non-neural data. It really didn't work that well. Once we put the neural data in, it worked a lot better. Okay, now important to some points to note. We are not using this technique to deny cochlear implant surgery. Okay, this is not for CI candidacy. Okay, we're really using it to augment decision making in terms of prescribing speech therapy and language and hearing therapy and music therapy. This is not to deny treatment. And in the current phase of our research, we're actually looking to see whether or not the predictive model can alter decision making in the dose of speech therapy that we prescribe to these children. And the research is ongoing. And this is our new cycle of NIH grant. And the project is conducted in the US. Okay, so very quickly, this is a Hong Kong project. Preterm infants. Okay, so from newer results from the literature, and certainly by now our local data, now we know that children who are born preterm compared to their full-term counterparts, their language and cognitive abilities are a little behind. Okay, so this is looking at using the CSBS, communication and symbolic behavioral scales, but we can use the Bailey, we can use the CDI. A convergence of evidence is now telling us that preterm infants, their language and cognitive development is not as good as their full-term peers. We have data from several hundred children now, at least 200 children now across different domains. We are also looking at children who are in different ranges of prematurity. So the moderate to late preterm versus the early preterm, even the moderate preterm infants, the moderate to late preterm infants, we're seeing differences, okay? And so the question is this: if we know that when the child is older, preterm infants' language and cognitive abilities as a group would be poorer than their full-term counterparts. But not everyone is like that. Can we collect some data early on and then construct predictive model to predict their language and cognitive development? Same kind of thinking, but now with preterm infants, okay? So we can use EEG. And in fact, EEG is actually pretty good in making that prediction, okay? So you can see that full-term infants, even if you can't read this, the signal is bigger here for full-term infants than preterm infants, okay? So like bigger, smaller. Bigger and more regular is better than smaller and not as regular. Okay, so you can kind of see that. So we can actually build these models. Our AUC is at as close to 90%. Okay, we can use that as prediction for prediction. Now we're also doing MRI on these children. So we have scanned over 300 babies in Hong Kong. Not everyone is successful. 200 some gave us useful data. The trick is we're scanning them unsedated. Okay, so it's very hard to try to get them not to cry when they're in a noisy environment. We made it happen, okay? So we've developed a pipeline, a scanning protocol, how to wrap them, how to like protect their hearing, like triple cover their ears. And then for some children, we put them in a scanner-compatible incubator because it filters out the noise a little bit. And then like this platform here makes the vibration not as strong. And so, you know, after throwing a lot of money away, basically, we learned how to do it. Okay, so we've gone through it over 300 children. Okay, 200 some useful data. Oh, I forgot to change this. This is now published. So we just published the first Chinese infant brain template and atlas based on the 200 and some, actually 100 and some from this template data that we collected. It's important to have a Chinese specific templates because the shape and the size of the brain is different than Western babies. Our paper actually is in NeuroImage made the demonstration that if we just rely on templates from the US, it doesn't work. So this is like the deviations, like the US templates deviated from the Chinese brains a lot, okay? And so that's why it's really important to have Chinese brain templates. So we published that. So with that, we can now really analyze the brains of preterm infants and term infants in details. So and then we look at their developmental trajectory across the 3 years of life. You can see that there's a linear trajectory across many different regions of the brain. And now we're waiting for more outcome data to come back, but we're using these data, the thickness and the volume of the different brain regions to construct predictive models about language and cognitive development. And they're working out pretty well. I mean, I don't have a full set of results yet because this is a longitudinal study. We're still collecting the data, but we've presented preliminary results in different platforms. So you can see like these auditory areas, cognitive areas, frontal lobe. Those are areas that are predictive of future language and cognitive development in these preterm children. Okay, so I'm not going to go through the results. Oh, okay, good. I'm right on time. Okay, so just quickly, the treatment for us, so we predict and then we prescribe. I mentioned that before. The prescription of intervention for our population, a lot of times is really teaching parents how to implement play strategies at home and communication strategies at home. In other words, how the parents work with their child is extremely important for our purpose. And so now we are using FNIRS and sometimes EEG to do interbrain coherence experiments to try to understand what's actually going on between the child and the parent as they interact. When we provide them with intervention, does that change that interbrain coherence dynamics? Okay, so we're doing some experiments like that. So this is actually published. There are various experiments. We just published a paper in Psychological Science concerning like how children who have autism interact with each other. And so we're doing these various experiments. Just want to let you know. And now, we presented this paper before, but now it's in Psychological Science. Okay, so that's all I have. As I said, the EEG test is now commercialized in Hong Kong. And I'm the founder of a startup company in Science Park. So I have financial interests and therefore I need to disclose those financial interests. So the research that I presented obviously is related to my company. That's why there's a conflict of interest and I need to declare that. And also the research that I presented is related to various granted patents and provisional patent applications that we have filed. So there's that I also need to declare. Okay, so to conclude, partly because of developmental disability, language and cognitive developmental outcomes can be predicted using data from even early infancy. Neurodata consistently provide the highest prediction precision on individual child level, not group level, individual child level. That, I have to emphasize that again and again, with or without non-neuroclinical data. Prediction can be made across a convergence of developmental outcome measures across different populations. And our preliminary data is suggesting that response to intervention can also be predicted. Neuroprediction, therefore, has the potential to inform decisions about prescribing different options of early intervention, even before a neurodevelopmental condition can be formally diagnosed. Having said that, I must say the following. We have to acknowledge the diversity and spectrum of developmental possibilities and respect an individual family's view about their child's neurodevelopmental condition. We also respect individual families' choices about what support services and intervention options that may be best for their child, given their own cultural context and viewpoints about developmental conditions. Some families believe that autism is a problem that needs to be treated. Some families don't. And so what we're presenting here, our Predict-to-Prescribe approach to neurodevelopmental condition, provides just one choice for families and is not meant to define all developmental conditions as a disorder requiring treatment for every child. So again, so some parents just don't think that they need to predict. Some parents think that autism is just fine. Some parents think that hearing impairment is just a culture and there's no need for any sort of intervention whatsoever. And here we will respect the choices the parents would make for their own child within their own cultural context. That's all I have. I have to acknowledge our different funders and also the large team of students and collaborators and postdocs and research assistants staff that have contributed to our research. So funding from Hong Kong and the US. So thank you.
Great. Thank you so much, Patrick. I think we do have some time for questions. I know I have some, but I'll let the audience go.
Yeah.
So much for the informative talk. So my question is for the predictive models. So the first study is about predicting language outcome, and the second one is about the responsiveness to intervention for children with ASD. So I'm thinking, so we see like really strong evidence for the EEG data, using EEG data, predict these two things. But my question here is, do we want to be more specific? So use a specific EEG
or specific populations?
Yeah, I mean for predicting different things.
Probably.
Yeah.
Yeah,
So
probably because our interest is language and cognition, and therefore speech processing makes sense because that's the precursor of more complex language, and so that, and then plus other research that have been done to build up to that. Yeah, I agree. So for different conditions and for motor, we might want to look at something else. Yeah.
Yeah, so following up with that question is, the age range. So from like infants to 24 months old, right? So 2 years. So do you see a pattern that data from older kids will have a better prediction?
Yeah, I understand your question, but the answer is no.
Okay.
From the data we analyze. So yeah, what do you think makes sense? When you're closer to reality, you're more accurate.
Right.
Because the reality is coming up when you're more distant from the reality. Yeah. So we did some analysis of the window of prediction. It really didn't really matter.
Yeah.
Again, that speaks to developmental stability. Yeah. And we also have developmental stability data on our EEG data, because for some subjects, we have multiple time points. They are stable.
Okay.
Yeah.
All right. Yeah. Yeah. That's good, actually.
Yeah.
Thanks.
Yes.
Thank you for this very nice talk. My question is like, have you ever looked at the relationship between FR response and gene or so I think you look at the relationship between the language skill and the gene for like one specific piece of gene, and that gene like not expand the language skill or predict the language skill. And you think like the data show like the EEG response and explain or can predict the language skill. But I think my question here is like, if the effort response can be determined by a specific gene, or is it determined by?
Yeah. So we some of these subjects, the 421 here, we also have the FFR data. As far as these genes are concerned, they are not related to the neuroencoding measures. But, we might not be looking at the right place because, it's the sensory nuclei. You should be probably looking at the glutamatergic genes. I mean, we're not, because the study is not designed to look at the function of that particular area of the brain. Now, in the future, if we have stronger hypotheses, we might conduct a study. But right now, I don't want to, spend all the resources just like fishing something that is hard to fish. But, we have some hypotheses about what kind of, or like where to look. Yeah.
I'm actually going to jump in here because my question was exactly about this analysis, which is, for background, one of my master's degrees is in statistics.
Okay.
So my question is this really a fair test for the genes, right? It's a regression main effects model, right?
Right.
And I don't know, this is not my field and genetics is not my expertise at all, but everything I've heard suggests gene to gene interaction effects, right? Which would suggest you'd want to do at least 2-way or 3-way interactions.
Right.
Please.
Yeah. So first of all, we're only looking at a very small set of genetic variants.
Yeah.
I think that needs to be taken seriously because there's a vast number of variants that we are not looking.
Yeah.
The point that I'm trying to make here is that for as far as we know, because we use a candidate gene approach, we're only looking at what we know because those genes have been looked at in some other studies. Otherwise, we'll spend a lot of energy fishing.
Yeah.
Okay. So I'm not making the point that genes has no effect. I'm just saying that as far as we know now, from our knowledge now, we don't have a model that can be clinically useful. Yes, gene to gene interactions, of course. These are these genes might be interacting themselves. In fact, FOXP2 must be related to FOXP. Now, but after some kind of filtering, we end up by having these because I think the models already consider overlapping variants. These are the only significant ones. Okay,
Right.
so something got kicked out, but not, no, we're not looking at interactions. In fact, we're not, no.
I just think, if I were you, I mean, you've got the data.
Yeah.
I would want to run something like a boosting algorithm on this, to see whether or not, I mean, it's exploratory, but yeah, you would just get a sense of whether or not looking at interactions would, because your argument here is that, hey, look, these are not explaining a lot of the variants.
Yeah.
Right. And that's fair. But, my argument is, okay, to play devil's advocate, you're not looking at interactions. And sometimes, those can have,
The stronger
interaction
effect.
alone, a huge effect. Yeah.
Oh, okay. Yeah. Okay. Yeah. Thank you.
Sure.
Yeah. Yeah. Thank you. Yeah. Good point. Yeah. Shelley. Yeah.
Your part, I think that's really big theory in the world. I wonder about whether you consider, whether the stimulating used for testing the neural commitment actually comparable to the original study because the actual study is segmental.
Yeah. Okay.
But for your next study, it focuses on superset.
Right.
Do you think it is fair to say actually neural commitment that?
Okay. We use what I did not present is some cortical data. Our cortical data replicated the neural PACU's results using segments. So when you look at the long latencies like MNN, it works. The neural commitment hypothesis works. So it's really about the brainstem. That's different. Yeah. Yeah.
Easy data to the language combination.
Yeah.
But what do you mean combination?
So it's just according to the Bailey. Yeah, so Bailey has the language domain and the cognitive domains, and then they ask you like this cause and effect tasks. No, the clinician directly assesses the child using the Bailey. So it's not a self-report. Yeah.
Not familiar with that, but I think they know from the most of cognitive work I learned is like basic combination.
Yeah, I know, like inhibition and working memory and all that. So the Bailey test is constructed around those constructs. Okay. And so like there are these like blocks that the child has to do, and then like you see one that the child would.
So that's also like a association.
Yeah, there is when they design the test, and Bailey is probably the most widely used developmental assessments in the world. I mean, there's like validations across different domains, but it's not looking at the nitty-gritty of cognition that we researchers typically would look at. We're not looking at inhibition. We're not looking at like divided attention, and we're not putting it in those boxes that cognitive scientists usually look at. Yeah, but it's based on those constructs. Yeah. Yeah.
I have two small questions. So for the lexical tone study, so did you mean even for the like neural encoding or entrainment, you didn't see the difference between the native tones and
Right, correct.
Oh my God. Okay.
We didn't see it in the FFR measures, the brainstem measures, cortical measures, yes.
Okay.
At 6 months, there's a dividing line with the cortical measure.
Okay.
Yeah, so then the child actually responded faster to the native tones than the non-native tones.
Okay.
That's what the neural commitment hypothesis will predict.
Yeah.
Yeah.
All right. So another small question is for this study.
Yeah.
So here, because you're predicting DSE, right?
Yeah, DSE. Yeah.
English. So one question here is the ICS. It's the most contribution here. But ICS sometimes is correlated with IQ. So do you want to parse out RQ first?
IQ didn't survive. We have IQ there.
Okay.
Yeah, IQ didn't survive.
Yeah. Thank you.
Interesting.
Yeah.
Yeah, thank you for the amazing talk. So I just wonder, like, I noticed in the fMRI setting, so basically the infants are sleeping in the sky, right?
Yeah.
So for the speech processing during sleep, so do you think,
It's all anatomical.
oh, just
Yeah, anatomical.
Okay.
Yeah.
Yeah.
And my personal suspicion is that the reason why EEG is doing so well in prediction is because we're making the child do something, like it's a
Yeah.
speech processing, not just based on the anatomy. So that's why the anatomy results are, if we do direct measurements down the road when we have enough data, we'll find that the EEG test will predict better than the anatomy. Yeah.
Okay.
With resting state, we all that too. But
Mm-hmm. So, but earlier in the presentation, so there are also like newborn falling asleep, hearing speech, right, in the new system.
majority of the infants were sleeping during the EEG paradigm. There's a minority of them, and we make note of that. A minority of them were awake. In fact, like 2 days ago when we tested one, I walked into the room, the child was like just on the controller and just like looking at, yeah. So that's a minority. Most of them prefer to sleep in the caregiver's lap, arms of their caregivers, which actually makes it harder to collect the data, but that's besides the point. Yeah, because the caregiver gets tired, not the child. Yeah, if the child is in a stroller, is actually a lot faster. Yeah. Yeah.
Professor Wang, thanks for your presentation. And I am very interested about your inner brain coherence part.
Yeah.
So I would like to ask, what is the meaning of the inner brain synchronization of the results? I mean, if they have really high inner brain coherence or low, what is the meaning about the results?
So what we're trying, that's exactly what we're trying to look at, is to see if interbrain coherence is plastic, and whether or not that plasticity is related to parental implementation of the intervention and ultimately the developmental outcome of the child, that kind of pathway. Yeah.
Okay, so
and then the other question has to do with the specific brain networks involved. Is the parents doing something to specifically enhance language, or the parents is doing something enhancing emotional and attentional mechanisms? And so that's why the brain results will be helpful.
Okay, so it's not only focused on the speech part, it's also involved the emotion and also the cognition across the parents and also the
Yeah, from the brain area. So we make connections about, like attentional processes with a specific brain network, with the channels that we look at, language processes being on the left. And, so we make hypotheses like that.
Okay,
Yeah.
thank you. So, and I also want to ask, like, when you analysis the inner brain coherence, did you just to analysis the each channel between the parent and also the children, or the region, or I mean, like, if you're gonna separate the different region of the brain and to analysis the data across the region, or for
So with FNIRS, and because we don't have each person's brain models, we're making some assumptions about the areas that we're looking at.
Okay.
And due to that, we have to do a lot of averaging across the channels.
Okay.
Yeah, across the optals. I mean, so there are standard practices that have been published in the literature about how to go about doing that, precisely because we don't really know where things are.
Okay,
Yeah.
thank you so much.
Thank you for talk. So I have a question, like, have you examined the association of the neural encoding of the multiple babies in one family? Because I was thinking, like, if I was a mom, I have a baby with neural encoding in my family, and now I'm going to give her birth again. Like, have any suggestion to me?
That's precisely why we have business. Is because in pediatric clinics, very often the parents would do the test is because their older child already has a neurodevelopmental condition. Yeah. And so, yes. Yeah, but yeah, that's going on already. Yeah.
Great. Other questions? In the back?
Hi, Professor. Can you hear me?
Yeah.
Hi, Professor. Thanks very much for an extremely interesting talk. I'm interested in in you being an academic, and then you in this area to do with hearing and things like that. does the university help you with, you know, your company, you know?
Mhm.
Are academic institutions supportive of people like yourself, you know, to go on this, you know, route of also commercializing what you have found? I mean, taking the step out of the academic.
Yeah. Are you in, what faculty are you in? Social science?
I'm in the law faculty.
Oh, in the law okay. So do you I don't know if you know, like how the government is really promoting innovations and technology. And so ITF, the Innovation Technology Fund,
Yeah.
has devoted millions and billions of dollars to help us do these things, like startup companies. And so the government has special money allocated to the university to help professors open up companies. So I took advantage of that and my colleagues. And then if you go to Science Park, for you guys, you can go to Cyberport. There are incubation programs for startup companies. They have funding, they have people to babysit you, to help you run your companies. And then you can apply for many different funding mechanisms from the ITF. That's like the culture and the ecosystem that the government is building up. And so it's a great time to be in Hong Kong, actually, if you're interested in that kind of route.
Thank you.
So we took advantage, completely took advantage of one scheme after another.
Oh,
Yeah. So, yeah.
thank you very
And
much.
students, like they're encouraging students to do it, and alum, and like, so students are coming together, and there's seed fundings for students to open up company as well. Yeah.
Thank
Like the tissue scheme, like you have it too here. Because all 8 institutions got money from the government. Yeah.
Fantastic. Thank you.
Great. Okay. Anyone else? Okay. If not, then let's thank Professor Wong.
Okay, thank you.